This conference has been recorded by Dr. Jacques Tremblay, Centre de Recherche du CHUL in Quebec City. It is about the development of a potential treatment for Friedreich Ataxia. All our tissue are made of cells which contain a nucleus, and this nucleus contains the genetic codes for every protein that cells make. There are several billion cells to make a human body.
Ceci est un enregistrement pour la diapo numéro 1. Cette conférence a été enregistrée par le Dr. Jacques Tremblay, Centre de recherche du CHUL. Elle porte sur le développement d'un traitement pour l'ataxie de Friedreich.
This is a diagram view of a cell. The cell membrane is represented in yellow. It is very thin. The nucleus is represented in blue, and it contains the DNA illustrated in black. The mitochondria are illustrated in blue.
C'est un test d'enregistrement pour la diapo numéro 3. On dit que les cellules sont bien belles, sont bleues, blanc, rouge, et c'est juste pour vérifier si ça fonctionne bien.
In cells, the DNA is organized in long strings, which are chromosomes. These chromosomes can only be seen when the cells are dividing. This is a picture of the 22 pairs of human chromosome and the sex chromosome X and Y obtained from a dividing cell. The pair of chromosome 9 is surrounded by a red square to indicate that these are the chromosomes where the frataxin genes are located. This is a cartoon view of a chromosome with the DNA double helix illustrated in it. In fact, in the chromosome, the double DNA helix is supercoiled, as we can see on the left side. On the right side, we can better see the double helix of DNA. In each chromosome, DNA is therefore a long double helix form of only 4 different nucleotides. The adenosine is always attached to a thymine. The cytosine is always attached to a guanine. There are 3.3 billion nucleotides that constitute the human chromosomes. During cell proliferation, this double helix DNA is separated in 2 strands, and each strand is copied again, the adenine attaching to a thymidine, the cytosine attaching to guanine, so that at the end of the cell duplication, 2 identical strands of DNA have been produced. Genes codes for protein. There are some sequence of DNA that codes for part of the protein. These sequences are called exons, and these exons are separated by sequence of DNA that do not code for an exon. They are called introns. Genes are made of a series of exons separated by introns. To make a protein, the double-stranded DNA has to be copied into a single-strand messenger RNA, where the thymidine is replaced by uridine. This single-strand messenger RNA is then exported outside the nucleus in the cytoplasm, where the protein will be synthesized. In the cytoplasm, the messenger RNA will be transcribed into a protein. There are 23 different amino acids that constitute these proteins. For each amino acid to be included in a protein, there is a transfer RNA that recognizes a 3-nucleotide sequence in the messenger RNA. This 3-nucleotide sequence is called a codon. In this cartoon view, we can see that there is a new transfer RNA that will become attached to the messenger RNA to add an additional alanine amino acid to the protein which is being synthesized. The DNA always has a double helix structure, whereas the protein, which are made of 23 different amino acids, have very variable shapes. In this cartoon view, we can see different proteins illustrated in green, orange, yellow, pink, blue. And as you can see, these different proteins have different shapes. Some of them are located across the cell membrane, which is represented in gray, and some are located only inside the cell, such as dystrophin protein, for example. The mutation responsible for Friedreich ataxia is located in intron 1 of the frataxin gene. Thus, the sequence coding for the amino acid of the frataxin protein are not modified by this mutation. In normal subject, there are 6 to 34 GAA repetition in intron 1, whereas in patient there are 150 to 1,700 GAA repetition in intron 1 of the frataxin gene. Nowadays, the entire human genome has been sequenced, and therefore it is very easy to go on internet and obtain the nucleotide sequence of any given human gene. This slide illustrates only a part of the normal frataxin gene. The sequence of nucleotide of only one of the strands in the DNA molecule are illustrated because the other strand is complementary to this one. This is part of the sequence of the normal dystrophin gene, and you can see that overlighted in blue there are 6 GAA repetition in intron 1. The frataxin protein is made of 210 amino acids. The sequence of these amino acids are represented in the center of this slide, each letter representing a different amino acid. In patient, as mentioned previously, this protein is not modified. However, it is less abundant because of the GAA repetition in intron 1. In the human genome, there are 2 frataxin genes, one located on each chromosome 9. If only one gene is mutated, the person will be an asymptomatic carrier of the disease. In the province of Quebec, 1 person out of 50 is a carrier of Friedreich ataxia. However, if both genes are mutated, the person will then develop the Friedreich ataxia disease. This is due to a reduction of frataxin protein in all tissue, including the brain and the heart. Frataxin play a very important role in mitochondria. This is simply a reminder that the frataxin genes located on both chromosome 9s are mutated in a Friedreich ataxia patient. The number of GAA repetition in intron 1 of the frataxin gene affects the severity of the disease. The longer are the GAA repeats, the less frataxin is produced, the disease starts younger and is more severe. Frataxin play an important role in mitochondria. Mitochondria are involved in the production of energy in the cells, and they contain iron. When frataxin is reduced, there is accumulation of iron in the mitochondria, and this produces oxidative stress, which lead to cell death. Different potential treatments are under investigation for the treatment of Friedreich ataxia. Each one of these potential treatments trying to respond to one of the pathology aspects. For example, there are expansion of GAA in intron 1 of frataxin gene, and this leads to a reduction of frataxin expression, which in fact may be due to a change of conformation of the DNA due to this long GAA repetition. So one of the potential treatment under investigation is inhibitors of histone deacetylase. Histones are proteins that are attaching to the DNA, and it is believed that by inhibiting the histone deacetylase, this will change the conformation of the DNA, and this will improve the expression of frataxin. A second aspect is the diminution of frataxin, which is being treated by administration of erythropoietin. The exact mechanism of erythropoietin action is not completely understood. There is also an accumulation of iron and sulfur inside the mitochondria. One potential treatment is to capture the iron with a drug called deferriprone. Finally, the accumulation of iron and the absence of frataxin in the mitochondria leads to oxidative stress, and therefore one of the potential treatment is to use an antioxidant drug, idebenone, commercialized under the name Catena. Several of these potential therapy are in fact targeting secondary problems of Friedreich ataxia, not the primary problem, which is a reduction of frataxin expression. Thus, the treatment that I am trying to develop is to administer to the patient the frataxin protein. This would restore all the role that the frataxin plays inside the cell. Similar approach of treating disease with protein have been developed by Genzyme to treat, for example, the Pompe disease with a protein called myozyme. Protein do not normally enter in the cell if they are injected simply in the blood of the patient. This is illustrated here in this experiment, where the beta-gal protein is normally not penetrating in the cells of the liver and the kidney. However, if this protein is coupled with a cell-penetrating peptide, for example, in this case, the TAT peptide, then the fusion protein called TAT beta-gal will enter the cell in the liver, kidney, lung, heart, and spleen, and even in the brain. And this gives the blue coloration that we can see on the right side of this slide. Therefore, for our experiment, we need to produce the frataxin protein. How do we do that? Well, we have to make a circle of DNA that is called a plasmid, and this circle of DNA will contain a sequence of DNA which is called a promoter. This is a sequence of DNA that controls when a gene, which is another sequence of DNA, will be expressed. So in this case we have a promoter which is illustrated in pink, and we have after this promoter the frataxin gene, which is made of 1189 nucleotide. This plasmid will then be introduced either into cells or into bacteria, and in both cases the DNA code will be transcribed into a messenger RNA, which will be translated into a protein in the cytoplasm. For our experiment, we have produced a modified frataxin protein. We see in red the sequence of amino acid in the immature frataxin protein. However, we have added amino acid to this protein. For example, we have added in green cis-histidine amino acid, which will be helpful for the production and the purification of the protein. We have also added in purple a sequence of amino acid called PEP1. This is again a cell-penetrating peptide similar to the TAT peptide used in the previous experiment. This will improve the penetration of the frataxin protein inside cell. We have also added to this protein a sequence of amino acid in blue, which is called the V5 tag, and this is simply to be able to detect the frataxin protein more easily in immunohistochemistry using antibody against this V5 tag. When the protein is produced either in cell or in bacteria, there is several other proteins which are produced also, and therefore we need to analyze this mixture of protein to verify whether our frataxin protein is really produced. The way to do that is to use a technique called gel electrophoresis. This gel is simply like a jello which is located between two glass plates. On the top of the gel there is some small wells into which the mixture of protein can be deposited. These small wells are then covered by a buffer, which is simply water mixed with some salt, and a negative electrode is deposited into this buffer. At the bottom of a gel, there is again another buffer with, this time, a plus electrode. When electric current is applied between the two electrodes, the protein will be attracted down this gel. However, the protein which are the smaller will move faster down the gel, and those that are big will remain on the top. After a few hours, if we stop this migration, we will have different bands that are the different protein which have been separated according to their size. This is an example of the analysis of the protein produced when the plasmid coding for frataxin is inserted into cell. In this case, the frataxin protein is detected with a specific antibody. We can see three important bands. The one in the middle is the immature frataxin protein, but when this protein migrates from the cytoplasm inside the mitochondria, this protein is truncated. It produces then the intermediate form. Finally, this protein is truncated again, producing a smaller protein called the mature frataxin. As you can see, this mature frataxin, being smaller, is located lower down the gel. As mentioned before, for our experiment, we have modified the frataxin protein by adding a cis-histidine tag and a PEP1 protein transduction domain. We were wondering if the presence of these two additional amino acid sequences in the frataxin protein would interfere with the maturation of the frataxin protein. The maturation, I remind you, was simply to cut the protein. So we have here on the right side an illustration of an experiment in which the plasmid coding for the cis-histidine PEP1 frataxin V5 has been added to a cell. we can see that after 18 hours, there is the presence of the immature frataxin protein, which is illustrated as PP, whereas after 48, 72, and 96 hours, we can see that there is more and more abundant mature frataxin protein, illustrated as PM. For our experiment, especially the experiment that will use mice which are a model of Friedreich Ataxia, we will need a lot of frataxin protein. We have therefore decided to produce the frataxin protein in bacteria. This slide illustrates bacteria which are growing on a gel. Each one of these little white spots is in fact a colony of bacteria already containing thousands of bacteria. This is a view of these bacteria at higher magnification. The bacteria that we use are called Escherichia coli, and they appear as small red rods in this illustration. The plasmid coding for frataxin is simply introduced into these bacteria. The bacteria will proliferate, and after a while we will sacrifice the bacteria, lyse them, and release the proteins, including the frataxin protein. This protein will then be purified and will be used for experiments such as injection into mouse. When we want to produce a lot of protein, instead of growing the bacteria on a gel as illustrated before, the bacteria can be grown in a liquid culture medium, such as the one illustrated here. We see 200 ml Erlenmeyer containing culture medium in which the bacteria are growing. For even a larger production of protein, the bacteria can be grown into bioreactors. Some bioreactors are as big as 20 liters or even 200 liters. So billions of bacteria can be grown, and these bioreactors are simply controlling the temperature of the mixture, the agitation, all the necessary ingredients to rapidly produce the bacteria. This is a gel electrophoresis analysis of the protein that are produced by bacteria. On the right side, we have bacteria which are not producing frataxin. As you can see, there are nevertheless lots of different bands that represent different bacterial protein. However, on the left side, we now have bacteria that are producing frataxin. And as you can see, there is a very intense band that appears in the middle of the gel. This is a frataxin protein produced by the bacteria. After the production by the bacteria of the cis-histidine TAT frataxin or the cis-histidine PEP1 frataxin, this protein has to be separated from the other bacterial protein. This is done by using an affinity column which contains nickel beads. This column is illustrated on the left side. On the right side, we have a cartoon view of this column, which contain the beads covered with nickels. They are illustrated in yellow, whereas the frataxin protein is illustrated in red. Due to the presence of the cis-histidine tag, these cis-histidine frataxin will attach to the nickel on the yellow beads. The other protein, which do not contain the cis-histidine tag, are illustrated in blue, green, and brown, and these proteins will simply flow through the column and be washed out of the column. This is an example of the analysis of protein which are produced by bacteria and are being purified. On the left side, label load 1, 2, and 3. This is a gel electrophoresis of the different protein produced by the bacteria. You can see that there are lots of different bands and therefore lots of different protein. Washing 1, 2, and 3 illustrates the protein that are coming out of the column when we are washing it. There is lots of protein coming out in washing 1, but after that, washing 2 and 3, there's no more protein coming out of the column. Then the frataxin protein, which is attached to the nickel bead in the column, can be eluted by adding a special chemical to the column, and then you can see that in column 2 to 10 there is this frataxin protein coming off the column, which it has been eluted, and you can see that there is only one band, and therefore now the protein is pure and it can be used for further experiment. This purified frataxin protein can be used for experiment with the cells of Friedreich Ataxia patient. These cells are grown in culture in vials illustrated on the right side. These vials contain a red culture medium which contain all the ingredients, the amino acid, the sugars, which are necessary for the proliferation of the cells. All of the manipulation of these cells in culture have to be done under a sterile hood because the culture medium used to grow the cell is also very good to grow bacteria, and at this point it is not the bacteria that we want to grow, it's only the cell. At the bottom left we have a picture of the cells which are growing in these culture vials. As mentioned before, the protein do not normally enter the cell from the culture medium. However, if the TAT beta-gal protein is added to human cells in culture, it will penetrate rapidly in these cells, and its presence can be revealed by the blue coloration illustrated here, showing that, again, protein coupled with TAT or with PEP1 will be able to enter cell. The same thing will happen when the frataxin protein is coupled either to TAT or to PEP1. It will enter the cell and it will eventually migrate into the mitochondria. Following the production of the frataxin protein, we have done experiments with cells which are very special cells obtained from a conditional knockout mouse model that has been provided to us by Dr. Puccio from France. These cells, as any other cells, contain two frataxin genes, except that one of the frataxin genes is already mutated. It contains a stop codon. I remind you that we have seen previously that the amino acid are coded by a sequence of 3 nucleotides. This is called a codon. Some of these codons, instead of coding for an amino acid, are coding for a stop signal. And when such a codon is encountered during the synthesis of frataxin, the synthesis of frataxin will stop at that point, and if this is a premature location, this will produce a smaller frataxin protein which is not functional. This is the case for the gene located on that chromosome. The other frataxin gene located on the other chromosome is a conditional knockout frataxin gene. That is, it contains additional DNA sequence which are called LOXP. There are 2 LOXP illustrated in dark blue in this illustration, and an additional sequence which is illustrated in orange. However, all of these additional sequences the ones in blue and the one in orange, all of them are a multiple of 3 nucleotide, and therefore the codons for the different amino acid are not changed after this sequence, and a frataxin protein will be produced, which is functional. However, when a gene coding for a special enzyme called Cre recombinase is introduced in these cells, one of the LOXP sequence and the sequence between the two LOXP will be removed. This will produce a different gene which now contains only one LOXP. However, this only one LOXP does not contain a multiple of 3 nucleotide, and therefore the codons coding for the different amino acid are changed after the LOXP, and soon you will encounter a stop codon due to the difference in the triplets, these codons which are being recognized. Therefore, the frataxin protein will end at this new stop codon, and again a non-functional frataxin protein is produced. Therefore, in these cells, following the action of the Cre recombinase, both genes are unable to produce a functional frataxin protein, and these cells will die very quickly. This is an illustration of the type of experiment that we have done with these cells coming from the conditional knockout mouse. On the top left, we have these cells which are growing normally because they are producing frataxin, and you can see that the bottom of the flask is completely covered with a layer of cell. However, if we introduce the gene coding for the Cre recombinase, the gene which was still functional, the frataxin gene which was still functional, is mutated, and it will no longer be able to produce frataxin. And within a week, all the cells are dead, as illustrated on the top, on the right side. However, if we add to the culture medium either cis-histidine PEP1 frataxin or cis-histidine TAT frataxin, then this protein added in the culture medium will enter in the cells which are not able to produce frataxin, and it will reach the mitochondria, and it will permit to these cells to function normally and survive, and you have lots of cells as illustrated at the bottom on the left side and on the right side. So this is a very important result because it shows that this recombinant PEP1 or TAT1 frataxin is able to enter the cell, and the frataxin play its normal roles and permit to the cell to survive. This is exactly what we want to do for the patient. In their body, we want to introduce these recombinant protein. They will enter the cells in their brain and in their heart, and it will permit to these cells to survive. And the survival of these cells will prevent the development of neurological and cardiac symptoms. Given that the previous experiment was extremely important, we have, of course, repeated it. We have obtained the same result. However, in this additional experiment, we have added an additional control. To one group of cells that were not able to produce frataxin, we have added to the culture medium the cis-histidine frataxin protein. This protein does not contain the TAT sequence or the PEP1 amino acid sequence, and therefore it is not able to enter the cell. And as you can see on the figure at the bottom on the left, there are very few cells which are surviving. It shows that the presence of the amino acid sequence in TAT or PEP1 are necessary for the protein to enter the cell and play its role. Since we started our project to develop a treatment of Friedreich Ataxia using TAT frataxin or PEP1 frataxin, there have been a few articles which have been published, including this one by a Korean group, Dr. CHUL. He has shown that the PEP1 frataxin was able to protect neurons from oxidative stress. This is a figure from Dr. Choi's article. It shows that the PEP1 frataxin has entered in the neurons in the brain of an hamster. On the left side, we have the absence of frataxin, and on the left side, we have this frataxin, this PEP1 frataxin, that has been injected in the blood of the animal and has reached into the brain, has entered into the neuron, and this gives the brown coloration illustrated on the right side. This is also a figure from the Choi article. It shows on the top rows of neurons that are dark blue. This is without ischemia. There's lots of neurons. When ischemia, a lack of oxygen, is induced, lots of neurons will die, as illustrated on the second row. However, if the hamsters have been previously injected with PEP1 frataxin, there are fewer neurons which are dying at either 4 days or 7 days after the treatment. And therefore, in row 3 and 4, there are much more neurons surviving than in row number 2. This Korean group has also published another article where they show that PEP1 frataxin significantly increase the cell proliferation of neuroblasts and their differentiation in the mouse dentate gyrus, which is a part of the brain. A Chinese group has also published an article where they have coupled the PEP1 peptide with copper zinc superoxide dismutase and with the catalase protein, and they have shown that this recombinant protein was able to enter in the cells of the heart, where it protected these cells from ischemia. Again, this is another example of recombinant protein, including PEP1, where the recombinant protein has entered into the cells and protected them. Finally, a very important paper has been published by the group of Mark Payne on November 23, 2011. This group has shown that the TAT frataxin protein increased the lifespan and the cardiac function of a conditional Friedreich Ataxia knockout model. In fact, this is the same mouse model as the cells that we have used in our experiment. This is a figure from the Mark Payne article. It shows that the TAT frataxin protein, labeled in green, has entered into the mitochondria, which are labeled in red. When the two pictures are superposed, it gives the yellow color indicating that the TAT frataxin protein has migrated into the mitochondria. This is also a figure from the article of Dr. Payne. It shows experiments that have been done with cells which are either able or not able to produce frataxin. It shows that when oxidative stress is induced by adding iron sulfur to the culture medium, the cells are dying. In fact, they have a very abnormal shape in figure 4. However, if TAT frataxin is added in figure 5 and 6, you can see that the cells have a much better shape and are surviving better in the presence of TAT frataxin. This figure is also from Dr. Payne's article. It shows the survivals of mice for 60 days. The normal mouse are illustrated in light blue at the top, and you can see that you have an horizontal line at 1 because all the mice are surviving up to 65 days. However, the mice which are developing the Friedreich Ataxia symptoms are dying very rapidly, as illustrated by the red line. You can see that by day 30, less than 50% of the mice are still surviving, and all of these mice are dead by day 52. However, if the mice are injected with TAT frataxin, they are surviving better, as illustrated by the dark blue line. You can see that at day 30, there is around 80% of the mice which are still surviving. At day 50, around 30% of the mice are still surviving, and some mice live up to 65 days. Thus, the administration of TAT frataxin has improved the survival of these mice and have for some time reduced the symptoms of Friedreich ataxia. You may wonder why the mice are dying anyway. Well, this is a very severe model of Friedreich ataxia. These mice are not producing frataxin in their heart starting during early embryogenesis, and therefore when these mice are born, their heart is already severely affected. This is the reason why these mice are dying as soon as 53 days. This is not the case in the patient that develop Friedreich ataxia. They are producing a lower amount of frataxin, but they are nevertheless producing it, and this is the reason why their symptoms are much less severe, and the symptoms begin only by the age of 20. So therefore, the administration of additional TAT frataxin will improve the survival of the cells in their heart and in their brain, and will thus reduce the progression of the Friedreich ataxia disease. Therefore, the results that have been obtained in Dr. Payne's group and in my groups are very encouraging for the potential development of a treatment based on the administration of TAT1 frataxin or PEP1 frataxin. However, before TAT frataxin or PEP1 frataxin can be moved into a clinical trial for Friedreich patient, a lot of additional experiments have to be done with cells and with animal models. We have to answer the question of whether the presence of frataxin prevents the accumulation of iron in mitochondria. We have to verify whether it increases the anti-oxidative enzymes such as copper zinc superoxide dismutase or kinetase. We have to verify if these recombinant protein re-establish the normal expression of all genes. This can be done with RNA-seq, a new technique. Experiments should also be done with a less severe model of Friedreich ataxia. That is, a mouse model which produces frataxin in lower amount, just as the Friedreich ataxia patient, but does not have a complete absence of frataxin, as in the experiment that have been done so far by Dr. Payne. We should verify whether the TAT frataxin or PEP1 frataxin enter the brain, the heart, and the muscles, and whether it reduces the symptoms. It is only when such results have been obtained in mice that we will be able to approach the Food and Drug Administration or Health Canada to have the permission to do clinical trials.